And the way to understand this file, here in this box we have our, and in this cell we have our windows for power to be at the N terminal, and we have in these two columns represent the sliding window. Remember how we set the sliding window of 15? So you'll notice that each of these are a sliding, they each have a length of 15. So it's taking basically an average of the properties in that window. And then these columns here represent the category totals. Remember 8 was the most different, and 1 was the least different. As we look down here, we have different z-scores. For example, here we the z-scores for each of the associated spans and each of the associated categories of magnitude. So the way that we read this is we're going to select all of the cells. So remember that it was significant in the categories 6 and 8. So we're going to select those cells. Let's first do, we can do 6 first. If you do Control Shift and then the down arrow, that'll select everything in that column. Then we can go up here to Conditional Formatting, highlight cells rules, greater than, and remember we're going to try and get everything significant at the 1/1000th level. So we're going to select cells that are greater than 3.09, and we can just have them be highlighted there. And as we scroll through these, we'll notice that there are some, for example, this patch right here is very significant, and that would be a place where we'd want to look into some more. Now let's do the same thing for our category 8. Control Shift down. And again, we have several patches where there was a category 8 change in this category. The last step to being able to understand this is we would like to have this kind of in a graph. Instead of having to scroll through here and trying to optically pick out where selection is occurring, we'd like to do this with a graph. So what we do is we select the categories that we want to graph. You can even graph the 4 or 5. You can graph the top 4 if you want, and just select them all the way down. And you can insert a line graph. Now, this is kind of hard to read, but basically we have a, well, we should label a series. So you can do this select data. And what it's saying is there's this series 1, which is this one on the far left, and just named it series 1. We can edit it and call that level 4. Change this to level 5. There we have our labels. So now on our graph, just generally down the very bottom, we can see that there is this level 8 is there's a peak right about here in this sequence, and there's a level 6 peak right around here. So you can see that while there's level Level 8 and level 6 were there. Level 5, which is red, did not show up in our z-score summaries table. Now the one thing to make this a little bit better, or there's things you can do to make this more readable. For example, you can shrink down the line a width, and you can make it more. And you can do that by, in this Format sub-tab, select in this drop-down one of the levels and Format Selection, and then the you can either change the line color if you want, the line style, you can turn on the width. Yes, I'm just going to go to 1 point here. So that made that a little bit more readable, and I'm just going to do that with all the rest of these. Alright, now that those lines are a little skinnier, it's a lot more readable. There's one last thing we can do to make this, I guess, more understandable. Right now there's a lot of action going on, and you can kind of see right now that there is levels of high conservation. For example, this area right in here, where there's not a lot of selection in any of the categories, would be an area where there would be high conservation. Also, this area right here would be high conservation, while areas such as this whole block in here would be under pressure, you could say. You can also do one more thing. In this vertical value axis, you can change this to have a fixed value of 3.0, and when that is done, it makes it very easy to see places where there is high variability and high selection. So for example, we have a category 8 right here, as well as some category 6s right in here, while the rest of the clutter is taken out. And that's basically a way to take our data from Mega and get out something useful, something where we can know that, where we can find conserved regions along the sequence. Now you'll notice also that these are the amino, or these are the nucleotide sequence, or these are the nucleotide numbers. So at the first nucleotide, that's right there. So if we wanted to map this onto a protein, we could either divide these by 3 to get to the amino acid count, and then plot that directly on there, or we could just line this up against the protein. And that's how to do TreeSAAP.